a lot of you. We also go out live on YouTube, and we usually have a few hundred people that watch the first few weeks after it goes out. We've got members that can't make it. Probably all our members of the OCA will rewatch, and then we get people all over the world watching over the coming weeks and months after our talks. Megan for getting that set up and providing this awesome service with all the great knowledge and content Master provides to the world. Got our presentation ready? Isn't that a beautiful fish? Hummingbird tetra, right?
Yeah.
See, I gotta get some now. You said that they're very sensitive.
Yep.
Touchy. Gotta really talk sweetly to them. All right. So Sudipta Shah, by the way, is a biochemistry researcher at the University of Minnesota in Minneapolis. And I don't have to give you this mic. I was going to, but you have your own. Is it all good?
Can you all hear me?
No.
No?
Turn it up. Little switch right on.
I see. I missed it. Is it good now?
Yes.
Okay. I can speak loud, so
Yes.
just let me know.
Project.
Yeah. Okay. Thank you so much, Steve, for the introduction. And thank you, Andre, for inviting me here. I'm really glad to be here. And today I will actually talk about how I grow plants in my tanks without injecting CO2. Some of the plants that are considered to be high-tech, hard to grow plants, I actually have been able to grow these plants for several years now without injecting CO2. So before I talk about my methods, let me just show you my first planted tank that I started in 2018 in Minnesota. This was a CO2 injected tank, so you can see the bubbles coming out there from the diffuser. But I was kind of like not that happy with certain plants. Some of the plants like Ludwigia Pantanal, I was consistently having problems growing it. But anyway, overall, the plants, at least in this photo, they look happy. Around the same time, I set up this 10-gallon tank primarily for my Caridina shrimps as a low-tech tank. I did put few plants in this tank, and to my surprise, most of them actually worked pretty well. I did put a lot of difficult plants as well in this tank, and they all kind of like did much better than some in my high-tech tank for some odd reason. So to further investigate that, I thought, why not set up another tank? So I set up this tank in 2019. So initially it was looking like that. I added a bunch of plants later on, and in few months it was looking like this. And then further on, it was looking like that. So you can actually see the date here, the year. And then something like this, then this, then this. So that's 2023, same tank. Started this in 2019. So almost 4 years, 3.5 years by this time. And the plants were doing pretty well. And some of the plants are quite difficult, like Ludwigia Pantanal and Meta here, Rotala tulunarensis, Amania pedicellata golden, Ludwigia senegalensis, and so on. There are many plants actually. And I was like, interesting. In those 4 years of time, I did set up a few more tanks just to kind of prove my point that it's not just one tank, okay? So I had this tank. I ran for more than 3 years. Similar setup, 10-gallon tank, nothing fancy. Another one, a 4-gallon tank, which I think I maintained for close to 3 years. And then this one, a 5-gallon shallow tank for almost 2 years. So all of these tanks without CO2 injection, okay? And as Steve mentioned, like many people said that it's impossible and I've been lying. So I kind of like went backwards basically, and kind of like as a researcher, I'm a biochemist. I kind of like investigate. I like to investigate things. Ooh, what just happened?
I don't know. It's an AV
It's
night.
It's the projector from
Oh,
above.
the other projector came on.
Yeah. Anyway,
Those plants look terrible.
I was like, what just happened to the tank?
Did you put saltwater lights on it?
So anyway, I think this slide would be fine with this light. So I actually put together these 9 factors, which, in my opinion, play a crucial role to actually have sustainable and nice growth of plants without CO2 injection for a longer time. And I will actually today talk about all of these factors as much as possible. Starting with CO2 availability in water. So CO2 is kind of interesting. Unlike oxygen and nitrogen, if you dissolve CO2, it can react to water, form carbonic acid, which can be further dissociated into bicarbonate, which can again dissociate to carbonate. And this process is pH dependent, as we can see here. So lower pH tends to have more CO2 and carbonic acid in that sense. Oh. Primarily primarily dissolved CO2, I mean. So, apparently, most plants, and particularly the colorful plants that we see in the hobby, aquatic plants, they tend to actually prefer softer water without higher carbonates. So that means that if we have a tank where we limit the presence of carbonate, we give the advantage to those kind of plants more than the plants that can actually take bicarbonate. So it turns out that almost 50% of the plants that we keep in our hobby, they can utilize bicarbonate as the source of carbon dioxide, basically as a source of carbon, I mean, because they can convert this bicarbonate to carbon dioxide and use it. They have this enzyme inside their cells. However, the colorful plants, they don't possess that enzyme, carbonic anhydrase, that enzyme is. And so they have to actually have CO2 basically present. And that's why when we actually inject CO2, you have noticed that it lowers the pH. And lowering the pH means basically now you are making more of that CO2 available for those plants. And that's why we can grow those nice colorful plants with CO2 injection, which are kind of difficult if we don't inject CO2. Another problem with bicarbonate is that bicarbonate is kind of like more energy intensive, so plants need to put more resources to capture that carbon source. And from atmospheric air, which I have heard from many people saying that it can contribute to 3 to 5 ppm of carbon dioxide, which I found kind of like interesting. It turns out that only less than 1 ppm carbon dioxide can come from the atmosphere. And I've done some digging and research, and I put the information here. You can actually go check. It's not possible to have more than 1 parts per million carbon dioxide at atmospheric pressure and temperature, basically. It's impossible from atmospheric air to actually have more than 1 ppm in water. Okay, so where does this CO2 coming from? Because we know that all plants need CO2, and I'm growing these plants, and I'm saying I'm not injecting CO2, and there's only 1 ppm or so coming from air, so it must be coming from somewhere, right? Otherwise, the plants won't grow. So it turns out that the CO2 is basically coming from the substrate. So this is one of my tanks, 4-gallon tank, and as you can see that I'm using kind of like a deep substrate level here, almost 2.5 inches in this particular tank. So I am using primarily soil-based commercial aqua soil. In this case, it was ADA Amazonia version 1. I have found that one to be the best. Unfortunately, they stopped producing that particular soil. So at this point, it's kind of like difficult to actually get a good quality aqua soil to have this kind of tank. So these kind of soil, they have plenty of advantages. First, they're rich in organic matter, which the microbes living in the substrate can decompose and form CO2. Another advantage of these soils is that they are kind of active substrate, so they can actually buffer the pH. And they buffer the pH in the lower region, like 5 to 6, which in my previous slide I mentioned that actually favors CO2 availability, basically, like having the CO2 in the form of CO2 gas rather than bicarbonate. So that actually helps a lot. And on top of that, because these are soil-based, organic matter-rich component, they have the nutrient holding capacity, CEC. So there are plenty of advantages having these kind of substrates. And generally what I do when I start my tank, I do this process called dark start method, where I start the filter, put water, run it for 2 to 4 weeks without having the light on, without any plants. After 2 to 4 weeks, I check the ammonia concentration. When it goes to 0.5 ppm or low, then I plant, and I plant heavy. And then I start putting light and do the regular maintenance thing and everything. And I tend to face less problems with algae and anything if I do this method. If I put plants straight up after setting up the tank, I tend to have more algae issues and things like that. Okay, let me show you the importance of substrate, like quality of substrate, how it actually plays a huge role. So this tank, a couple of years ago, I set up, and as you can see, this was after dark start method. I planted pretty heavy. A few weeks later, the tank was looking like this, decent growth. But then in less than 2 months, the tank converted to this. Similar method, I didn't do anything different, and pretty much all of the plants stopped growing. And I was doing exactly what I did with all of my previous tanks. So I was adding the required amount of fertilizer, doing water change, everything. The only thing that I was not doing was CO2 injection, which I have not done in the other tanks as well. So the difference is the soil, which this time I used a different brand. So this particular one, I never tried this one before. I tried it, and as you can see, it didn't work that well.
Do you remember the brand?
This one, if I remember correctly, it's called Scaper Soil. I forgot the brand. It's from Germany, something like that, yeah. These type of soil, like pretty much every substrate is going to work out well with CO2 injection. Without CO2 injection, that's the trick here, is like you need to have good substrate. Okay, moving on, water parameters. Initially, I was using remineralized RODI water, remineralizing with calcium, magnesium salts. Then I switched to acidified tap water. So basically, I'm just now adding acid to my tap water before adding to my tank. So I won't recommend this method to anyone here, like unless if you know basically how much acid, and like if you know how to deal with acid, then do it. Otherwise, remineralized RODI water would be the best option. Carbonate hardness, almost 0. GH, between 5 to 7. And TDS, close to 150, something like that. pH in most of my tanks tend to be between 5 to 6. You can see this one at that point was reading 5.15. Okay, now light. This is kind of little, I think, I don't know, like at least initially when I posted this on Facebook or something, people were saying like it's not possible actually. Like I was putting that much light that people thought that it's impossible to have that much light without CO2 injection. How much light? I can show the exact number, 150 par at the base level in this non-CO2 injected tank. Most people will struggle with this level of light even with CO2 injection. Not all, but many people I have seen struggling with high light even with CO2 injection. This one, 155 par at the bottom. This one, also 150. This one, at this crowded place, this is a 20 high tank. Even in this crowded place, it's like above 100, 120. So that's like substantially higher amount of light than that what people actually recommend putting in typical low-tech tanks, like substantially higher amount of light. And it was like people would say, like, what's the advantage? Why put so much light? And my argument is that when you put high light, it can penetrate deeper, as we can see here. That means the entire plant, like even the leaves at the bottom, can actually get access to good quality light, right? That means that these kind of systems are already limited to CO2 availability because we are not injecting CO2 in these systems. And then our argument is that because we are not injecting CO2, we don't need that much light. But my argument is, if you are not injecting CO2, the plant is basically trying to get as much CO2 possible, so it's spending its energy, its resources, its enzymes production for CO2 capture. And at the same time, the same plant, we are asking it to grow well with low light. That means it has to now share some of that energy that it was using for CO2 capture for light capture. So it's just one plant, but it cannot do both. So by putting enough light, I have eliminated that part, so plant doesn't need to be concerned about availability of light. So it's putting most of its resources for CO2 capture rather than trying to put energy to capture light. So that's like one of the hypotheses around this. There is another actual scientific reason. It's about the enzyme that captures the light. So it turns out that that enzyme needs to be activated, and that activation is light dependent. So I'm not going to go deep into that, but trust me, high light does not cause algae, and high light is not bad for low-tech tanks. As long as you do the things properly, algae is not going to be a problem in these type of tanks. Okay, nutrients. This, I have found that many hobbyists put a lot of emphasis. In my opinion, without CO2 injection, if you are using good quality substrate, you don't need that much nutrient. And as you can see, this is the amount that I am basically putting in my tank weekly. That's pretty low NPK amount and some micronutrients. However, I do put ammonia-rich root tabs under the substrate from time to time. So I won't put the root tabs of fertilizer in the beginning, but after 2 months or 3 months, something like that, I would start putting root tabs occasionally. And that tend to kind of like do more benefit than having the water column rich in nutrients. Okay, flow and filtration. I generally use HOB filters. Let me see if I can actually play this. As you can see, it has decent flow and also the surface agitation that also kind of like helps for gas exchange. And another thing what flow does is basically it disrupts something called the boundary layer. So think of boundary layer as a thin layer of fluid in case of aquatic plant that is slightly different in its property to the overall water. So that means if CO2 or nutrient has to go needs to go inside the leaf or the surface of the leaf, it has to penetrate that thin layer that's slightly different in property. So having decent amount of flow helps to actually break that boundary layer so that CO2 or other nutrients can actually penetrate the leaf better. And this is why this, like when for CO2 injected tanks, people recommend a decent amount of flow, 5 to 10 times flow. So actually that helps to break the boundary layer and for the plants to get the nutrients. Okay. Then plant husbandry and maintenance, that's another very important factor. And as you can see, the root mass under one of my tanks. So I generally trim the tops of my stem plants, and I try to actually, I try to avoid frequent uprooting of stem plants. If I have to uproot plants, I would uproot from one side of the tank this week, and then plant, and then maybe next week or 2 weeks later, uproot the second half. I won't uproot the entire tank like what most people do with CO2 injected tank. There are several reasons for that. Is that when you have this much root under the substrate, it's doing many things actually. So plants, when they photosynthesize, they kind of like produce a lot of oxygen, and a lot of oxygen actually goes through the roots inside the substrate. And in the substrates is where the microbes are living, the beneficial microbes that are converting the organic matter to CO2. And those microbes need oxygen to do that process. And roots basically deliver the oxygen right to them. So if you are frequently uprooting the plants, that means your substrate is not saturated with roots. And that means the part of substrate where there is not enough root mass, it's going to get choked basically, and that means it's going to have situation of like anaerobic conditions where instead of having beneficial bacteria, you would start to support the bacteria that actually harm the ecosystem. So it's going to go the other way. And the other thing is that many plants can actually uptake CO2 from their roots, such as Vallisneria and many species of Ludwigia. So there are many benefits of having dense root inside the substrate. Okay, a few things about husbandry is that I generally do 40 to 80% water change every week. Whenever I do water change, I gently clean the substrate using a turkey baster and a hose. And I also do occasional deep cleaning. So whenever I have to uproot certain plants or bunch, that area I would do a deep vacuum cleaning. That's going to basically rotate the substrate. Basically, the bottom level of substrate is going to go on the top, and it's basically going to kind of like circulate the nutrients in that area. And also filter cleaning, I tend to do every 1 to 2 months, something like that. This is probably not that important in that sense. And also what really helps if you want to actually maintain these kind of setups for longer time is that if you can actually kind of like replenish some of the older aqua soil with new. So basically take out 10% or something like that out and put 10% fresh on top of it. So nothing fancy, maybe every 8 months or a year. If we do that, we can actually maintain these kind of tanks for several years without actually like disrupting the entire tank or something. Let me show you what actually happens if there is a lapse in husbandry and when the tank actually gets super old. So it becomes like that. The same tank that I showed you, that's exact date. So this happened when I went to India for almost 6 weeks. I asked my friend who never kept fish before to feed my tank, you know, the like few nanofish you can see. I don't know if you can see from distance, but there are like particles here, like these are fluval bug bites. When I came, I saw like there was just like hanging all over the tank. And this time, this was like 3.5 years old tank, and the substrate got compacted pretty well at that point. Even though I was doing all of those methods that I was suggesting, still it happened. So all of my plants were covered in algae, and I talked to some of my friends and they said, you know, just like it's been 3.5 years, just get rid of everything and, you know, start fresh. And I was like, nope, I'm going to revive this tank. And what I basically did, unfortunately, I don't have the exact photo, but I kind of did something what's going on here. I chucked all those plants, at least the top, to the shrimp tank, and they kind of like cleaned just like the sponge here in 2 hours or something like that, the top. I planted them back in the tank. For the wood, which was covered in BBA, I did spot treatment with freshly prepared mix of 3% hydrogen peroxide and Excel, Sechem Excel. Then I also did deep cleaning this time because I had to uproot all of them, all of the plants together. I deep cleaned the entire tank at once, and also thoroughly cleaned the filter, did like large water change, and planted those tops that my shrimps cleaned. And in a few weeks, this was looking like that. And then it was looking like that. And then like this. In less than a month, the same tank has like come back to life basically. So I did not do anything special here. All I did just like basic husbandry, and it took care of itself, you know. Okay, temperature. This is one factor that I have seen in hobby that very few people talk about. And as a biochemist, I never understood this. It's like they talk about everything except temperature. So temperature is like, it's I don't know, it's like when I do experiments, I have to maintain certain temperature. Otherwise, the reaction is not going to actually perform well, or it's going to be like faster or slower. So solubility of gas, regardless of which gas, is basically inversely proportional to the temperature. So the higher the temperature, lower is the solubility. So that means when we increase the temperature by 10 degrees Celsius, all biological reactions double in that 10 degrees Celsius increase. And it is vice versa. You decrease the temperature by 10 degrees, it's going to slow down by half. And most plants, and especially in my tank, they actually do much better is at this temperature, 70 to 75 degree Fahrenheit. If I go to like 80, I think I can still manage it, but beyond 80, it gets really difficult. And below 70, it gets kind of like super cool, so it gets slow, and most fishes don't like temperature that cold. So this is a kind of like a sweet spot that I would say that actually works pretty well. And yeah, that's what I was mentioning. Winter, plants do much better in my tank compared to summer. And one thing I've noticed during summer is that if I do a frequent but small water change compared to one large water change by the end of the week, it does much better in terms of plant health to actually maintain good growth even at high temperature. So I would say like 10 to 20% of water change, if you can do every other day or maybe every day, when the temperature is super high, that actually helps a lot. Okay, moving on, microbial diversity. So this is another thing that I have not seen many people talk about. In the hobby, all I see people talking about nitrification process, which I'm not denying that it's not an important factor. However, it's not just it's not the only process that's happening in the tank. There are many things happening in our aquarium because it's a tiny ecosystem. So what I did basically, I collected some samples from my tank, from my substrate, from my filter, and I isolated DNA from it. And I am kind of lucky in that respect that in my lab, there are people who do soil microbiome analysis. So I threw some of my samples with their samples, and there is a PhD student who did analysis of my tanks. And let me show you just one sample from one small area of my tank, just about bacteria. This is like what I found. So people talk about two bacteria, one Nitrosomonas, one Nitrobacter. That's the, yeah.
How much DNA did you also have that was not identified?
Oh, probably, I don't know, like 80% or something like that, because
That 600 species is only 20% of the bacteria.
something like that, something like that.
So we're actually talking about 5 times that.
Probably,
1,000
yeah.
species of bacteria. That's just bacteria,
This is just bacteria.
not eukaryotes.
Not eukaryote. I did eukaryote analysis in the second sequencing, and we got fungi. And then with bacteria, we can get archaea as well. So we got archaea as well. So I can talk about that after my talk, when I finish my talk. But there's some fancy things happening in the tank. So in short, a healthy tank is going to have a ton of diversity, not just 1 or 2 or 3 or 4 types of bacteria. It's going to have hundreds of types of bacteria. That's the, it's not about, that's why I didn't kind of like make this big enough for you to kind of read. It's like, oh, I have this bacteria, that. everything is important. It's an ecosystem, okay? So higher the diversity, healthier the tank is gonna be. And that's the main factor here. It's not about just one or two or three types of bacteria, okay? So basically, I went through all of these in kind of like brief. There are like a lot of other details that I don't think I have time to actually discuss here. But overall, these 9 factors that I have discussed today, if we can actually kind of like think about these factors and how to optimize these, then it's not just me. I think anyone here can actually get these kind of growth, you know. And I'm not just saying, I have actually proof of that. Let me just show you. This is not me, Travis, a well-known aquarist in Minnesota. He was able to replicate my method, you know. So it's not just me. Like that's another thing that like someone said that, oh, it's just you know, you have a degree in biochemistry, you're an expert, you can do that. I was like, nope, it's not just me. Other people can do this. Steve, another well-known aquascaper in Minnesota, 2 tanks, you know, pretty nice growth. Rakesh, he's my lab mate, and I'm kind of fortunate to actually have him in my lab. We have been discussing about this for the last 5 years. And he was the one who actually suggested me to put those difficult plants in my non-CO2 tank, because I saw in YouTube that those plants don't like with a tank without CO2, you know, so I was never even trying those. He said like, you know, you have so many plants, why not just try that? So I started putting those plants and noticed that they were actually doing kind of better, some of the plants, better in my non-CO2 compared to my CO2 tank. And he has actually multiple tanks, so that's one of them. That's another one. He's growing HC Cuba here. You know, that's one of the kind of like difficult carpet plants to grow in a CO2 tank. This picture he sent me last week. So, you know, like it's absolutely possible to have this kind of this kind of growth. Anyone can do this, you know. Vin Kuti, I'm pretty sure some of you might know him. He is quite well known in the hobby to actually bringing some fancy new plants from all over the world. So he tried this and it worked well for him as well, you know. So yeah, before I end my talk, I just want to kind of like quickly show you some of the plants that I love growing in these setups. Amania pedicellata golden. This plant is one of the most difficult plants in the hobby. Even for seasoned aquarists, you know, it's not easy to maintain this kind of growth for longer period of time. For maybe a month or two, fine, but then it's gonna like have those like distorted growth, especially on the top. And these kind of setups, it's so easy actually most of the time that I can grow in a small cup. This is a 600 milliliter cup, like that can only, it can only hold 600 milliliter of water. And you can see that the plant is doing fine. Another variety, Gracilis mini, Amania, and Senegalensis. These are also quite difficult, but in these setups, they do fine. Rotala tulunarensis, a fairly new plant in the hobby. Yeah, this one is from India. This is also a little difficult to grow, but these setups, it does pretty well. Oops, I think I skipped one. Rotala wallichii, Rotala rotundifolia HRA. These are slightly easier plants in that regard, but nonetheless, they're beautiful plants. Ludwigia senegalensis, another plant that I really like, you know, because of its mosaic pattern on the leaf. This one also does pretty well. Ludwigia inclinata, verticillata varieties, all 3 of them, at least the one that's available in the hobby, Cuba, Pantanal, Meta, all 3 grow just fine in these tanks. And then Cuphea utriculosa, it's not that common to have this plant. It's kind of like probably the most difficult plant, I would say, the ones that I showed. But this one, surprisingly, does really well in these kind of setups, you know. So yeah, that's pretty much what I have for today. You know, I have my journal at plantedtank.net. You know, you can actually find a lot of information there. I also have a journal, but probably plantedtank.net has more information. So ScapeCrunch, you'll find some information. Also have a lot of photos at Flickr. You can go and actually kind of see the photos itself. So thank you very much for your attendance.
Have you ever tried the white whale plants, the ones that are usually required, that people talk about requiring 45 parts per million of CO2, like Hygrophila chai or blood vomit? Yes, he has.
Blood vomit, I tried. I don't think I have picture or anything. I have a lot of pictures, so where is it? Where is it? Somewhere. Oh boy. Let me see. I am sure I have the chai. See if you can notice anywhere. I don't know. I might have deleted the photo. I have tried chai actually. Chai worked fine actually. I have Ludwigia white right now. Unfortunately, I don't have a picture here. I have on my phone, I can show you. Ludwigia white is doing fine. I tried Aeroquin. I was able to actually keep it alive for 3 months, but after 3 months it died. But regarding the first point you mentioned about the 40, 50 ppm, first of all, I'm going to disagree on that point that those plants need that ppm CO2. My argument is that you have Ludwigia Pantanal, which I showed, right? Well, where is it? This one. Between Ludwigia Pantanal and Aeroquin, which one do you think grows faster? Pantanal. Okay, then if Ludwigia Pantanal is that grows faster, that means it's using more CO2, right? So if this plant is using more CO2 and I can grow this plant relatively easily in these kind of setups, why do you think a slower growing plant would need 45 ppm of CO2? If a plant is growing slow, why would it need 45 ppm of CO2? It doesn't need 45 ppm of CO2 in my opinion. It's less efficient in grabbing CO2. It's not using CO2. When we put that much CO2, basically it's probably using 2 parts per million. So it's less efficient. It's not that it's actually using it.
Right, yeah. It's definitely not using it at all.
Yeah.
I will say for some of those plants, I definitely failed at the classic 30 to 35, and then have to crank it up to 45 to get it there.
Yeah.
But you make it work with less.
Aeroculum Sulawesi, that's another plant that I can grow pretty easily in this. I haven't been able to grow the quin, but Sulawesi, really easy plant. Yeah.
Yeah, I had 2 questions. The first one was, could you do the same thing with sand-capped organic potting soil, which is aqua soil? And then secondly, I noticed that most of your tanks are minimally hardscaped. Is that a requirement, or do you just not hardscape your tanks and more Dutch style?
So first question, the sand cap organic, it's absolutely possible. I have done that. The shrimp tank actually, the one with the yellow shrimp, that is the one that I'm running like that with organic matter at the bottom. Or it's a potting mix, and then I capped with sand. And I've been able to keep it for really long time. I think that tank I've been running, I still have that tank for 5 years now. The problem with those kind of tanks is that if you are running or trying to grow these type of plants which require lower pH, that means you kind of like those tanks, there is no buffering agent, right? Buffering substrate. So that means if you want to keep that low pH, 5 years of time, that tank is going to crash. It's not going to maintain that low pH for that long. It will probably maintain for a year or two. After that, it's going to be much difficult. So you need to have something to buffer the pH in those kind of setups. Okay? And the second question, I forgot. Hardscape. Actually, the tank that I showed, the 20-gallon with algae, that has some wood. I have at the bottom, actually, the lava rock and something else, I forgot. But I won't recommend putting any stone or anything that can actually add KH. I know Sirius stone, it leaches carbonates in the water. So those kind of hardscapes, that is not going to work in these setups. They will constantly increase KH of the water, and that's basically kind of like just going to go through the buffering, like the buffering capacity of the soil will be exhausted in less than 6 months. So otherwise, there is no reason to not have hardscape. We can have wood, we can have lava rocks, we can have dragonstone, those kind of, and I think there are few stones. I don't do much of hardscape, so I'm not that like familiar with all the names. But there are some rocks that are kind of neutral, that don't leach carbonates. So those kind of hardscapes, definitely, we can do that. Yeah.
I was gonna say, I was gonna bounce off your thing, Andre, talking about the plants that they talk about 45. Could it be possible those plants are more, they need more from the root rather than in the water? So the idea could be that even if you're increasing the amount of available CO2 in the water, what they're taking up would be more better taken through the root rather than through the water.
That's a good point, and that's why I tried the Quinn, and it actually kind of worked for 3 months for me. But after 3 months, it did not survive. So there are many plants that can actually uptake CO2 from roots, but probably not Quinn, Erioquin. There are other plants that can do that. I think Sagittaria can do that, Vallisneria. There are many Ludwigia species that can do that, but I'm not sure if Quinn can do that. Yeah.
Which Ludwigia species do you know of that are taking CO2 carbonate from the soil?
Not carbonate, CO2. If I remember correctly, it was one of the Ludwigia Rubin or Repen, something like that. I have the paper saved somewhere. I can send it to you. Yeah, they did some pretty fancy experiment. They kind of like separated the root zone with from the shoot zone, and so that they made sure that it's basically the top half is like they completely eliminated CO2, right? And so the only way to get is from the substrate, and the plants they were growing. So yeah.
So you don't suggest using sand on the bottom of the tank at all, any of the plants, or sand?
You can absolutely do that. What I'm saying is that then some of the plants that I showed might not do that well. There are other plants that will work. If you're talking about like you mentioned about the organic substrate on top and then sand, then yes, absolutely, it's possible. Yeah, but just sand, if you're asking just sand without any organic matter, that's going to be a little difficult to actually have this kind of growth. Yeah. Say it again. Well, that's the thing I've been searching. Like as I mentioned, ADA Amazonia version 1. I found that that's the best one, right? I tried at least 7 or 8 different varieties, and none of them gave that kind of performance. I haven't tried the new Feast from Dennis Wong, so that's one thing you can try, I have seen people, like my friend Rakesh, like the last photo I showed, one of the tanks, he's using Fluval Stratum. So that's probably a decent choice, I would say. Yeah.
You mentioned the dark cycling that you do.
Mm-hmm.
Can you speak a little bit more about that?
Yeah.
I've used in saltwater, but I don't know if
So-
freshwater hobbyists do it.
So basically, it's not, I won't say it's a method. All I do basically, I put substrate, put water, put the filter, and let it run without the light. No plants. Let it run for 2 to 4 weeks. I check the ammonia. When I see the ammonia level close to 0.5 ppm or less than 0.5 ppm, then I actually do a big water change, put plants, then put water, and just do regular cleanup, maintenance cycle. So I do not actually start with low light. I start with the amount of light that I showed. So I don't dim my lights. So, yeah.
Do you have a brand of light that you recommend?
Well, depends on what is your preference. Like, if you want that vivid colors, then there are several brands right now. I think Chihiros, I use mainly Chihiros. Then there is Weak Aqua, that's another brand. And all of these brands are Chinese brands, I haven't tried Twin Star. I have heard good things about that brand. I haven't tried that. But my point is, it's not about the brand. It's just about the light itself, I can grow, let me quickly pull up this photo if I can do it. This one, this jar, the light on top of this is a grow light from Home Depot. So it's not about the brand. Any light would do it as long as it's good enough, the par. It's about the par, the energy. So even a tube light like this is going to work pretty well, actually. It might not look that well, but the plants would be fine because they're getting enough energy. So, yeah.
I wanted to go back to the sand-capped organic substrate for a second. So compared to using an aqua soil, can you get the same results using a sand-capped organic substrate and putting a bag of aqua soil in your hang back or in the canister?
I have not tried that method. I have thought about it. It will be interesting, actually. You can probably put that in a canister. It's going to buffer the water. Yeah, that's a good idea. It is definitely possible, yeah. But one issue with that method, regardless of how you want to do it, is that whenever you have to uproot some plant, it's going to be a little messy. So if you can deal with that, it's not a problem. I just said that, like, I have one of those tanks for more than 5 years at this point. But it doesn't grow the best plants, in terms of color and vibrance, but it is still working. Yeah. I guess that's it then. Thank you.